Tonight's topic is forensic anthropology. Our speaker is John Jay College professor Angelique Corthals, a member of our Department of Sciences. Dr. Corthals will be speaking about the use of exciting new forensic and genetic technologies to gain fresh insights into ancient civilizations and to create an historical ecology of disease and biology so as to understand the transmission of disease and population migration patterns. Very importantly for us in the 21st century, the period Dr. Corthals has studied and will discuss this evening was a time of widespread environmental change, including climate fluctuation. And we all know firsthand that we are perhaps again in another such time period. So this research and the application of the scientific findings of forensic anthropologists like Dr. Corthals to our situation today may help us to manage our own environmental changes and challenges. Before we hear from Professor Corthals, I would like to introduce our moderator for the evening, Dr. Phyllis Della-Latta. Dr. Della-Latta is the director of Clinical Microbiology Service in the Division of Infectious Diseases at Columbia University and a professor of Clinical Pathology in Medicine at Columbia University Medical Center. She is a recipient of the Distinguished Achievements in Clinical Microbiology Award given by the New York City branch of the American Society for Microbiology and is a fellow of the American Academy of Microbiology. I give you Dr. Phyllis Della-Latta, who will introduce Dr. Corthals.
Well, thank you so much. I have a distinct pleasure to be here for the first time. I was born and bred here in New York City. And I've seen this building, and I've seen it's very pretty, very beautiful, and I thought you were training policemen. So I had no idea that this exciting field exists. And the introduction, thank you very, very much, by the way, Provost Jane Bowers. And also thanks to Anastasia for inviting me. Okay, and my life is my bio, and my little summary is kind of boring in comparison to the speaker because she has an exciting life, and it's a field that, if you're all entering this field, is really exciting. I mean, you hear about her summary. She not only is a distinguished forensic scientist, but she also does anthropology work, and she works in areas of infectious diseases, autoimmune disease, and she bridges the gap from way back in ancient times to the present day. Her work is focused on human and non-human remains with forensic purposes in pathogenesis of autoimmune diseases and infectious diseases. So that kind of bridges the gap between us right now, because my main focus is to detecting infectious diseases and pathogens and specimens and kind of detective work in that angle. But yours is far more in-depth and far more interesting in many ways. I think it's exciting. I mean, I absolutely love her field, and it's opened up a whole new world. And thank you very much for bringing interdisciplinary fields together. So join me in a nice warm welcome for Dr. Angelique Corthals.
Thank you so much for that amazing introduction. I don't know if I'm going to be up to it, but it was. Thank you very much also for inviting me, Provost Bauer and Anastasia. Thank you so much, and thank you to all my colleagues who are showing up. Thanks. It's always nice, and the students. That's right. Don't make me laugh during the talk. So, yes, there will be a lot of mummies in this talk. Be glad about that. It's interesting because mummies really have a great entrance into the imagination of people. You either think of it as something incredibly eerie and creepy, or you think of it as something incredibly interesting that could actually unlock a whole lot of mysteries. And thanks to modern technology, we actually can do that. Those dead really do tell tales. Modern technology is allowing us to unlock a series of information that we couldn't have done earlier in the history of science. So this is actually the story of how we use this technology and how it unlocks mysteries that can be useful nowadays that I'm going to talk to you about today. So who is that mummy? Well, how did it start? It started with an anonymous mummy, a mummy that had been found actually by Howard Carter. So it takes us back. Howard Carter is the discoverer of the famous Tutankhamun, right? Yes. So here's a satellite picture of the Valley of the Kings, and you can see that KV60 is the red dot that's surrounded by a circle, and you have another tomb called KV20 right next door, and KV20 is actually the eponymous tomb of Queen Hatshepsut. What does it mean, eponymous? It means that it actually contained the name of Queen Hatshepsut. It actually said, this is the tomb of Queen Hatshepsut. KV60, on the other hand, was a tiny, tiny little tomb dug out in real haste with no writing whatsoever. And that's why the mummy was completely anonymous, because not only did it lack a sarcophagus, but also it lacked writing around it that could potentially identify it. But you see how close KV60 is from KV20. Not just that, those two tombs are put at a very strategic place in the Valley of the Kings, as they are right on the cliff that's above the temple of Queen Hatshepsut herself, which is that structure that you see on the right-hand side for you, on the right-hand side of the picture. Now, those are part of the list of clues. As you can imagine, these are very loose clues. It's like, okay, so we have the tomb of Queen Hatshepsut, and we have as KV20, there's no bodies in KV20, even though the sarcophagus is there, there's no mummies in it. And then we have a tomb hastily dug nearby that contains two female bodies. Okay, but still a very tenuous clue. So the second clue that we got was this little box that you see. And that little box was found in a cache tomb. And the cache tomb that it came from is TT320, which is right at the bottom of that temple of Hatshepsut. And that cache tomb was actually a tomb which had been, again, dug out in great haste way in the late period of the Egyptian Empire, at a time where the Valley of the Kings was raided by tomb robbers. Because yes, tombs in the Valley of the Kings were already being robbed in ancient times. Whenever times were tough, people would actually take to tomb robbings because there was a lot of gold in those tombs. So these tombs, these hastily dug out tombs, were actually used to hide away the mummies of the royals so that it wouldn't be destroyed, because of course mummies contain a lot of gold amulets within the bandages. So what the tomb robbers would do is that they would literally saw off every limb of the mummy to actually retrieve those gold amulets, which is a kind of a grim job. So part of the material that was saved was that little box, and that little box actually has the name of Queen Hatshepsut. And what do you think that little box is? Jewelry. It could be a jewelry. Organs. Yes, absolutely. So it's a canopic box, which means it's a box in which the organ of the queen would have been put during mummifications. As you know, when the Egyptian mummified, they took out all of the internal organs. They very smartly actually realized that the first thing to go when a body is decomposing are the soft organs, in particular the liver. And actually, this is exactly what happened with this. This is the box that supposedly contains the liver of the queen. One of the first objects that was CT scanned during the inquiry was that little box that I was telling you about that had the liver of the queen. So what we were expecting to see was, you know, a massive blob of liver, and that was about it, but we just thought it could have something in it. And lo and behold, it actually had a white mass, and that white mass was a tooth. So now you have a tooth in a box that has the name of a chepsut. What are you going to do? What? That's right. You're going to actually try to match the tooth to the mummy that you found. So the next step was to actually take a CT scan of the mummy, and lo and behold, that mummy was missing the exact same tooth that was inside the box. Why was it missing? Well, because you can see that, and this is my slide this is a slide that I always show to students to say, brush your teeth, because this is what's going to happen if you don't. A chepsut suffered a massive tooth abscess, dental abscess. And it was so bad that it's probably what actually killed her. The abscess went into her sinus cavity and she died of sepsis. By the way, it can still happen to you. So soon as you have an abscess, take care of it, right? The tooth was perfectly fitted to the mouth of KV68. However, in our day and age of scientific skepticism, that wasn't enough. We needed to find out whether we could relate this mummy to other mummies of her family members that were kept at the Museum of Egyptian Civilization in Cairo. And so we went on to the molecular analysis, and this is the point at which I joined the actual project. So the first thing to do was, of course, when you extract DNA from a mummy, it's just the DNA of that mummy. You need a comparison DNA to match the relatives up. If you can relate that mummy to other known relatives of that mummy, then you have a match. Then it's very likely that it really is the mummy of a chepsut. And so we went up into the cellars of the Egyptian Museum in Cairo, which is a place unto itself, which is a really scary place, actually, where all the great discoveries are stored. Most of what you see in that museum, which is already incredible, is actually 2% of what they have in the cellars. And so we looked up for the relatives of a chepsut. We looked up for first her great-grandmother, Ahmose Nefertari. And you see me looking at her very, very closely here on the right. The reason why I'm so close to the mummy is not because I'm myopic, but it's actually because I was smelling her. I know, I like to smell mummies. What can I say? But the reason why I was smelling her is because she had a wonderful smell of cumin. And that was the part of the spices that were used to embalm her, because she was a royal mummy. And then we looked for the mummy of her father, Thutmose I. Then we had to poke holes into those incredibly precious mummies, which was incredibly stressful. So the task at this point was to train the team, optimize the protocols, and perform the pilot study to generate the preliminary data that will tell us or not whether this is the mummy of a chepsut or not. So the way we devised the protocol is to look for two types of DNA, the nuclear DNA and the mitochondrial DNA. So the first try that we did was to recover the nuclear DNA. And it's very difficult to recover nuclear DNA for ancient samples. And the reason being that the DNA in ancient sample is extremely damaged. But on top of that, you only have one copy of that nuclear DNA per cell. So it becomes extremely challenging to retrieve a full gamut of that DNA that can be sufficient to actually tell any type of stories. And so at the time when we were devising this, ABI was testing a new kit, a new forensic kit that's actually now a standard in forensic, which is a kit called the Amplificer. And what that kit does is that it shows you, it doesn't actually sequence the DNA. So you don't see ATGC, blah, 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 blah. What you see is a pattern of repeat of nucleotides. So what you see is really a pattern more than you see the letters. And if the pattern between the two mummies match, then you have a relative match. So you know that that mummy is related to that other mummy, right? So we were very successful actually with this, despite the fact that it's difficult, but the kit allowed us to actually extract the mummy of Hatshepsut successfully. And then, not wanting to rest on our laurels, we also extracted the much more easy mitochondrial DNA. Why is it easier to have mitochondrial DNA? Well, because mitochondrial DNA is found in multi-copies inside the cells. But the problem with mitochondrial DNA is that it only reveals one side of your family history, only the maternal side, because mitochondrial DNA is only passed from mothers to offspring. It's not very good at determining the degree of parentage between two people. So, you only have like a percentage of parentage between two people. And so for Hatshepsut, it was not the best, but we really wanted to use mitochondrial DNA as a control, because if we couldn't retrieve mitochondrial DNA, which is easier to retrieve, but on the other hand could retrieve the nuclear DNA, which is harder to retrieve, then that meant we had a problem with the nuclear DNA. It probably was contamination and we should redo everything all over again. But we did get nuclear mitochondrial DNA, and as a matter of fact, it is actually the mitochondrial DNA that gave us the first clue that indeed the mummy seemed to be the mummy that of Hatshepsut. And those results actually have now been corroborated by the second lab, which has been built in the meantime in Cairo, where they've performed an independent analysis, and that's currently being published. So this shows you the process of going from this body and the many clues of that body and down to the molecular analysis, which gives you an additional I don't like to call it proof, because we don't try to prove things in science, but at least an additional way of not discarding the fact that this mummy was going to be the mummy of Hatshepsut. Like the way I put that in very, very secure terms. So that is the work for Hatshepsut. Now, the other work that I do is actually a lot more lugubrious, but it's great for Halloween. It's called the historical ecology of infectious disease. So the infectious disease, as you all know, is about identifying what pathogen is responsible for which disease. Like here you have Plasmodium falciparum and Vivax, which are the two main species of malaria. One is cerebral, the other one is a common malaria. HIV is responsible for AIDS, and Mycobacterium tuberculosis is responsible for tuberculosis. So identifying the pathogen. And so what we did with my students is that we started the DNA amplification of the actual pathogen that causes tuberculosis. So what we have here is an oasis, which is very secluded, but also a center of migration and cattle ranching at the same time. It's like the perfect storm for tuberculosis. You have cattle, which are non-transmitter of tuberculosis, and a population that migrate back and forth. This was a really good example of how we could connect the dots in history and actually use that to predict where new focus or foci of a tuberculosis would occur. And you can do that with, other pathogen too. You can use that with malaria, and you can use that with the plague just as well. So to conclude, I just want to say that it sounds really like almost like a dilettante occupation to look at mummy DNA. But it really isn't, because it actually has application in everyday life. So from the mummy work that I did on Hatshepsut, ABI finally commercialized that test that we actually tested for that mummy work, which proved that the test was efficient and that it was very, very good for damaged specimens. So now most of the DNA forensic lab will actually use this little kit. So that's another application of this, at first, dilettante kind of hobby. And that's how I'm concluding. Thank you very much for listening.
Good evening, everybody. My name is Lawrence Kobylinski. I'm a chairman of the Department of Sciences at John Jay College of Criminal Justice, and Angelique is one of our star faculty. Actually, all of my faculty are stars in different disciplines. I was very interested in bringing a forensic anthropologist to come and join and round out our staff. We cover all kinds of sub-disciplines within forensic science. And I started to learn about Angelique's background and experiences, and I'm just so happy that she joined us. And of course, she's on leave now at Stony Brook. What a lecture, what an amazing talk. I took some notes, and I can't believe how many different things you covered, from shotgun proteomics, migrating populations, stable isotopes, TB, plague, don't sleep with dogs, fleas. I mean, it's just an amazing presentation, so I want to thank you. We're going to take a couple of moments just to have our speaker and moderator take a chair, and we're going to open up for Q&A.
I have a question that I thought I might interject here. In working out outbreaks and epidemics in the hospital, which unfortunately we do have, although we don't like to admit we do. We have a little investigation, epidemiological investigations. But we are then able to sort of fingerprint different strains of organisms. So if we have a MRSA, methicillin-resistant Staph aureus strain, and it's from a nurse, a doctor, and a neonate or a couple of neonates, we want to know if it's the same clone, the same strain. If it is, then we know how the spread of that particular clone went, or if it's just a garden variety of MRSA, which is a breakdown in infection control. Now, my question to you is, you're doing all this excellent work with actually kind of identifying what went on with the different queens. I'm not going to remember the names, the queens or the eras, also, and the kings. Right. Do you do any kind of typing to see who's related to who? Can you do that sort of fingerprinting in terms of a molecular basis with the humans? Because I'm dealing with little prokaryotes.
Oh yeah.
And not eukaryotes.
Yeah.
So can you do that to figure out how related one would be with another?
Yes, absolutely. You can do that, but you can't do that accurately with what the type of DNA that I talked earlier with mitochondrial DNA, but you can definitely do that using SNPs, which are single nucleotide polymorphism, which is, for those of you who don't know what that means, it's just one letter inside one of those nucleotides, or one of those building blocks of DNA along your entire nuclear genome. So on all of your chromosomes, right? So it's a series of those letters that shows you a pattern for a particular individual. And that, so the kit that we use, the ABI kit that we use, is actually designed to do exactly that. So it has a series of loci for those particular letters, and if person A has loci A, B, C, not D, not E, but F, and person B has A, B, C, not D, not E, and F, then chances are they are related. But, and here we have an ancient sample, so sometimes those loci just are not recoverable, and so we don't get the full answer. So we will give you, again, a confidence interval of how confident are we that these two people are related, but it can never be 100% confident on the relationship between the two people.
Just tell us who you are and where you're from, please.
My name is Takesh. I'm in a master's program in forensic science here at John Jay, and before I came here, I was at a graduate school of medicine at the University of Tokyo. And I was wondering how much the Asian DNA are damaged, because I thought if you could look at the DNA modification, like the tumor suppressor gene P53 is methylated, maybe you can tell he was or she was suffering from cancer.
Oh, yes, you're a P53 guy, huh? Yeah, technically you could. I mean, if you work on if you have enough of a sample that you can retrieve that particular part of the genome that will give you the information, yes. You can also try to use proteomics, actually, to look at P53 itself. And that would also give you a good indication. But you should always couple that, and that's the big lesson of doing this kind of work, is that you shouldn't limit yourself to one technique. You should always use another technique that will back up or that will not back up your findings. So in the case of the mummies, I did DNA proteomics and CT scans to bring the picture together. But yeah, with P53, you're stretching. You're stretching our science, but it's always good, because when we can stretch the science, that's when actually we make great advances. But yes.
Any other questions? Well, hearing none, I want to thank both of our speakers. It's just a fabulous presentation. We don't just teach police, we teach science. This is really a growing science college. We're very proud of it, and we're very proud of Angelique for her presentation today. If you are new to John Jay, come back and visit us. We will give you a tour of the facilities. It's just great. And thank you so much for giving a just fabulous presentation. We thank both of you.